Okay, if I draw the pathway, so starting from here, because that is where the buffer is, then actually it enters the paper towel, and then of course it is go through the whole system. See, when it is go through the system, so what is on the glass plate, right, is the paper towel. What is on the paper towel is the it is a gel, right? So gel, sorry, is a gel. Gel is on glass plate. Glass plate on 2 bricks. And you can see the paper is coming out, touching the basing here, touching the buffer level, absorbing the buffer like this. And on top this glass, right? So you have that paper towel. See, there is one of the major layer. Like this one. Right? Make sense? Now it goes to just so on top of that, we put our gel. Right? Our gel. On the gel, we have nitrocellulose paper. Right? So it go through nitrocellulose paper. So it go through the gel and it go through nitrocellulose paper as well. So when that happens, right, the DNA, imagine the DNA in this well, because somebody going through them. See? So when it is going through them, this DNA in this well, DNA embedded in the gel, come out, go through nitrocellulose membrane, and it will give an imprint on the nitrocellulose paper. It will give an imprint on the nitrocellulose paper, right? So this is the gel, probably. Yeah. So after gel, yeah. So the when it is going through the gel and then going through the nitrocellulose paper, so all the gel pattern is right on the nitrocellulose paper. Get the imprint of all the DNA. So DNA moving out of the gel and go through the nitrocellulose paper. When they're going through the nitrocellulose paper grab all the DNA. So as a result, you get exactly the pattern that was in your agarose gel. Now same pattern repeated in the nitrocellulose paper. Exactly what it is. Right? So now the DNA banding everything in the nitrocellulose paper. So then what you're going to do? So you bake it. Baking means you heat it in an oven, centigrade 17 degrees for some time, right? Because of the salty water, this DNA is already single-stranded, right? Break open or break the hydrogen bonds and they open. So, and because of the nitrocellulose paper, they immobilize. Immobilize means they can't move. They're firmly on the spot, right? When that happens, you get this nitrocellulose paper. You get this nitrocellulose paper. It has a single-stranded DNA. In the banding pattern is now on this nitrocellulose paper. All good? Up to that, understood, Devdu? That's fine. Up to that.
Yes, sir.
So yeah, then what we do? The PCR probe. We put the PCR probe, right? So that is, but before we put it, we have to heat them and make single-stranded. So the single-stranded PCR probe come and bind to single-stranded band, the band which have the insert. Okay, so then you get a band which has the insert. So now you know, right? It's a radioactive band. Therefore, you can detect exactly where it is. So now this is the marker, right? Marker is in here. With respect to marker, this is lane number 1. Lane number 2 also have the same band. It's there. So remember, the probe is only binding to your insert. They're not binding to other area. So then you can locate the mark on the nitrocellulose paper. You take that nitrocellulose paper and you have to have another gel. So new gel, not the previous one is finished now. New gel have the same pattern, and you take the nitrocellulose paper in front of the new gel, and you identify exactly where the DNA, right? Where the DNA is located, right? By nitrocellulose paper will give the exact location, right? So then you run a new gel, right? And new gel you find which fragment is the one having my gene, having the insulin gene, which fragment. So then you have number of ways. All the ways have the same DNA. Then all the fragment together, see, together. They all the same fragment in all the ways have the same gene, So then you cut this one physically with the blade. I show you how it is done, right? Physically, you cut and separate. So you can see this lady is cutting the band in the gel. See, cutting it from the gel. Yeah, so that's how you isolate them. So then you put into, right, special kit we call mini pep kit. So in that kit you have a filter here, the blue color one filter. Actually this band with agarose, so if you put it that before you put into this one, you take a separate test tube and put the cut fragment in there, and you heat a bit. When you heat it centigrade say 70, the agarose gel dissolves or melts, and then put a bit of water there, and it the DNA come to the water, right? So then you send it through this filter by suction, or either by putting another tube underneath, and do a bit of centrifugation, right? So then all the solution go through the filter, and filter does not let all the other impurities to come in, right? Only the insert is collecting in here, right? So is that clear? So how did you isolate the insert? Right. So maybe you can put this video again, and if you have any doubt, you can find it out, right? I keep going, otherwise we're running out of time. So then I go back to the same place where we come from, right? So we talk about separation of the fragment, gel electrophoresis, identifying the DNA piece, insert using DNA probe, right? Only that is they told. But what technique you use, you can say it is Western, southern blot, right? So you can say the DNA probe made from PCR fragment, right? So running a gel and then cutting the small fragment and isolate from filtration, right? So those steps you can put in between. Now then they're asking selection of bacterium. Now we're going to clone them, right? So we have to find a suitable vector and a suitable bacterium host, right? Suitable bacterium host. We call it a host cell, right? So we're going to clone. We're going to insert this. We're going to insert this small plasmid vector into the bacterial cell, right? So that if the bacteria are not matching, the bacteria will destroy the plasmid. Not all the plasmid mobilize in all the bacteria. So you need to identify the suitable host, right? So you can't put certain vectors or certain plasmid in different bacteria like salmonella or like different species of bacteria. But you have to put it in. If it is E. coli base, then E. coli let it to have. Otherwise they break it down, right? So how do the cells identify their own DNA and foreign DNA? Yeah. So can you remember we got methylation, methylated fragments? I think second class we done that bit of that. So methylation. So bacteria know that it is a foreign DNA, or even our cells know that whether it is a foreign DNA or whether it is a self DNA. Self DNA they identify by methylation. Methylation process is adding small methyl group CH4 groups onto the DNA, right? But the way they adding that is different from each other. So this pattern will recognize, bacteria can recognize, or any cell can recognize their own DNA and foreign DNA. If it is foreign DNA, they destroy. Therefore, so in my projects, like so before you put the insert even, it is a good idea to make the vector, right, or make the vector inside the host cell, right? Like yeah, you have to make the plasmid vector, right? But when you extracting the plasmid vector, right? So it has to be the same cell where it come from. If it is come from E. coli, and you get E. coli to host it, right? So that's what that step you understand now, right? So you can mention that selecting suitable bacteria and suitable plasmid. 2 steps already, right? So then they explain the some characteristics of vector, right? So example, antibiotic resistant gene,